Hello friends, welcome back to my channel Medicine of 101. Today we'll talk about type 2 hypersensitivity. Okay, so before starting the topic, let me tell you, I have already uploaded the video in introductions part in terms of the normal hypersensitivity, their types, their classification as far as their extent is concerned. After that, I did upload the video based on type 1 hypersensitivity, where is 2. Okay, now I'll talk about today type 2 hypersensitivity. As I mentioned in the introductions part of the hypersensitivity videos, that basically we'll get what the type 2 hypersensitivity is mediated hypersensitivity. Okay, so first thing we'll get it over here, that this is the antibody mediated. Okay, and basically in these cases we'll get the antibodies IgG, or you can get the IgM antibodies. Okay, and in this we get the fixed intrinsic antigens. What type of the antigens we'll get over here? If we just talk in terms of the antigens, get fixed intrinsic antigen. Okay, these antigens, it may be the self antigens, or we can get the self antigen that can be modified. Okay, we can get write it over and modified self antigen. So I'll give the examples, then it will be more clear for you. Modified self antigen. Okay, now let me tell you first. Okay, for example, if we this is the RBC over here. Okay, now on the RBC you can get, on the surface of the RBC you can get the antigens over here. Is the self antigens of the RBCs, against which we can make also sometimes the auto antibodies. You can get the auto antibodies. Okay, or for examples, the same RBC we are getting over here. Okay, now see, we are getting the, let me show over here, self antigen is the self antigen of the RBC. Now for whatever any reasons, if we just say for any kind of the drug is coming over here, after coming the drug, it is modifying the antigen. For example, the penicillin, we can say antibiotics. Okay, so after coming there, we can say it is the modified antigen, against what we can get the antibodies. We can get the antibodies. So we can say in these cases that we will get the antibody. We'll get the antibody, this antibody against cell surface. Okay, or you can get extracellular matrix. On extracellular matrix here, you can say extracellular matrix. Okay, this is the normal thing. Now the type 2 hypersensitivity we can class in the three ways. So we can see the type 2 hypersensitivity we can get in the three ways. Okay, so now I'll show you what are the classifications of the type 2 hypersensitivity we can get. Okay, first let me erase this part, then I'm going to discuss the each individual types. Or the, okay, let's, now as far as the classifications of the type 2 hypersensitivity is concerned, we can classify in the class A, can classify in terms of class B, we can classify in terms of type or class C over here. Okay, now what type of thing we'll get in these cases? Let's talk about first class A. In these cases we'll get target cell depletions. In these cases we'll get the antibody that will be produced to, or to what, to diminish or to do the depletions or to destroy the target cell. So first we'll write it over here, target cell depletions or destruction. Target cell depletion or destructions, definitely antibody mediated. Okay, first we'll talk about this part, then we'll go to the subsequent parts. Okay, first let's talk about the target cell depletions or destructions, that is definitely antibody mediated. Now in these cases, if we just take the example, for example what I took first, the RBC. So okay, let's take in this way, the RBC over here, and now we got, there is the antigens over here, the antigens over here, the antigens over here, the antigens over here. Now against which we will get the autoantibodies. The autoantibodies, they will come over here. Okay. Now the autoantibodies, that means, for example, here the IgG, they are having the certain kinds of the receptors over here. Okay. Then some certain types of the receptors for different types of the cells. For example, if I see over here on top of the cell, okay, let's make in this way. So this kind of cell is over here, that is having the what receptors on what? On the antibodies. These are the receptors. Now whenever this receptor that will activate this type of the cells, that will produce the different intracellular signals for phagocytosis. Then what are the thing we'll see? This, try to understand. These cells, for example, there is the macrophage over here. For example, I'm just saying the macrophage. This macrophage that will engulf the what? That will engulf this whole antigens over here. As a result, we will see whatever the thing is over here. This antibody is getting attached with the antigens along also with the cells over here. So this type of the action that is known as opsonin. The first things we can get under the class of target cell mediated, or we can say the target cell depletions or the destructions antibody mediated without inflammation. So the first thing we got over here, that there will be no inflammations. Okay, first the antibody will come. This antibody is having the receptors for certain types of the cells. These cells that will go under the phagocytosis process, that will engulf the antigens over here. Now this type of the action is known as opsonin. So opsonin means, in these cases we will see the IgG that will act as opsonin over here. While the IgG on side that is getting attached with the antigens, another side of the IgG that is getting attached with the phagocytic cells. As a result, it will help the phagocytic cells to engulf the antigens, over here. Second type of the thing, if we just say over here, for example, this is the RBC over here. Okay, let's talk a little bit. Okay. This is the RBC over here, and we are getting the different kinds of the antigens, surface antigens over here. And against what we are getting, the productions of the autoantibodies. And from the complement systems, I think you know it, that whenever the FC regions of the complement system, this is the FC portions of the complement system that will be activated, that can stimulate the different kinds of the what? Complementary proteins. First we will get what? C1, then we will get what? We will get the C4, we will get, then what we will get? We will get C2, then we will get C3b, and we will get C5b, C6, C7, C8, C9, just like this. Okay. Now think about this thing. This C3b, for example, we have to talk in terms of the C3b. For example, this is the antigens over here. Let's make in this way. This is the cell, for example. Okay. Now this C3b that can get detached from here, and this C3b is coming over here in these regions. This is the C3b. Now there are certain types of the cells, phagocytic cells, they are having the C3b receptors. Okay, there is a presence of C3b receptors for certain kinds of the cells, phagocytic cells. Now in the same way we will see over here, again we will get another types of the what? Phagocytic cells that will come over here, and that will engulf the antigens. Now the C3b here, that is getting attached where? One part is attaching with the what? Antigen, another part is attaching with the cells. So now the C3b will get the option in here, the IgG. First I already did talk about it. Then we will get also the C3b. So C3b will get it over here. In terms of that will also work as what? The C3b that will also work as opsonin. First we got the IgG, antibodies. Then we got the C3b molecules over here. Now we'll talk about another type of the thing over here, that under the target cell depletions without inflammation, The next thing we can talk about over here, that for example is RBC over here. Okay, let's drawing which part. Okay, I'm just erasing this part over here. Okay, then I'll have space to go to the next part. So opsonin we got for the first thing we got in terms of the target cell depletion, the opsonin where we got IgG and C3b. Now we'll talk about another types for it. For these things. Okay, now in this case try to understand. We'll get the antigens, surface antigens. We got the surface antigens. Now against the surface antigens we'll get what? We'll get the productions of the autoantibodies. And we know whenever in autoantibodies they will be activated, we know the Fc regions of the autoantibody that will be activated, for example over here, and whenever it will be activated, it can stimulate towards certain kinds of the complements. For example, it will come C1, then we will get C4, then we will get C2, then we will get C3b, then we will get C5 convertage for it. It will just make the C5 convertage that will attach what? C5b, we will get C6, C7, C8 and C9. Okay, now from C5b to C9, try to understand from C5b to C9, that will form the on top of the thing that is known as membrane attack complex. So the C5b to C9 that will form the membrane attack complex, and this type of the thing is seen just like this. If we see, it will form a channel just like this thing. This is the membrane attack complex. Now, whenever the membrane attack complex will come, whenever it will be formed, it will come where within the cell membrane of the RBC over here, and that will just that will work as a drill machines, or so that will just do the lysis of the cell. As a result, we can say, let me show over here. Now, after the formations of the what? After the formations of the MAC, that will come over here. Okay, and that will form the drills, that will form the pore. As a result, we will see whenever the pore that will be formed, definitely there will be what? Osmotic imbalance. So we'll get what? Osmotic. This is intercellular thing that will go outside, and whatever outside that will come inside. So we'll get osmotic imbalance. So osmotic, we'll get what? Osmotic imbalance. So whenever we are getting the osmotic imbalance, the cell will get die for the osmotic shock. There will be what? Osmotic. We get the osmotic shock. As a result, the cell that will get died over here. Okay. So this type of thing we can also see in terms of the target cell depletions or destructions, antibody mediated without inflammations. We got first the opsonins. Then second class we got what? For the permissions of the MAC. That means membrane attack complex. That is for involving of the complement systems. So from C5b to C9 that will form the membrane attack complex. Then we'll get this other thing. Now what type of the thing we'll get the next steps? Another type of we can get over here. That is known as, okay, let me write it over here first. So yes, let's erase this part. So class A is not completed yet. I'll continue in class A, then we'll go to class B. Now the last thing about the class A, we can see over here, for example, is the RBC. Okay, it's not coming clearly. Okay, let's draw it once more. Okay, in this case, try to understand from here. Okay. Now, for examples, we know that antigens are present over here, and antibodies is coming over here. Within the antibodies, okay, within the antibodies, there are certain types of the receptors that will come in contact with certain types of cells directly. So there will be some types of the cells that will come in contact with these things, because these cells are having this type of the receptors. Now these cells that will come over here, and that will cause the lysis of these cells. That will cause the lysis of the cells. We can see the what type of this thing will get over here. That is known as antibody-dependent. Okay, let me write it over here. Antibody-dependent. Okay. Cell-mediated cytosis, cell mediated cytosis. So we can write it over ADCC in this way. So antibody dependent cell mediated cytosis. In these cases we will see a cell to come into contact. So cell contact, for the cell contact with the antibodies, these cells that will just do the what? Kill, that will just do the killing of the cells. That means in terms of the antigens over here, in terms of RBC. Now this type of the cells, it can be the neutrophil, it can be the macrophage. Okay, this type of the cells we can get over here. So in this way we will get what? That the contact cell killer. We can say write it over here, contact cell killer. Okay. Now this type of thing we'll get in terms of the class A. So in the class A, what type of the thing we got it over here? First we'll write once more in this cases, then we'll go to the different parts, different class. Now in the first part we got over here, okay, let's write. First we got the opsonin related, opsonin that can cause the damage of the cells or the antigens, engulfing of the antigens by the phagocytosis process, that will help to or continue the phagocytosis process. So in these cases we got the IgG, we got the C3b complement proteins. The next one we got, the second type we got what? The formation of the MAC, that is membrane complex from C5b to C9. That will form what? Membrane complex, and for the osmotic shock, we'll get the date of the antigens related cell over here. Okay, the cell with antigens. The third thing we got it over here, antibody-dependent cell-mediated cytosis. Okay, write it over. Antibody-dependent cell-mediated cytosis. Okay. So in these cases we got, there may be the neutrophil, there may be macrophage that can come over here. And whatever the thing we can see in terms of the cell, target cell depletions, where can we see? We can get in terms of autoimmune hemolytic anemia, OM type. OM type means this antibody they are able to work under the normal body temperatures. Then we can get autoimmune thrombocytopenia. Okay. Then we can get ABO incompatibility. That means by chance if I am A positive, if I just get the blood from the B positive, then whatever the problem I will face, try to understand. If I A positive, if I get the blood from the from the donor over here, B positive blood, then whatever there will be productions of the auto antibodies that will come over and that will cause the lysis of the IBC. So in this cases, so in case of the ABO incompatibility, we can get this type of the problem. So we can get also here in terms of erythroblastosis fetalis is one of the most important thing. Erythroblastosis fetalis. In these cases we can get. In these cases, whenever the Rh negative mother and Rh positive fetus. In this case, the Rh negative mother that will produce the anti-Rh antibody against the fetus Rh. Okay, and after that we will get what? This antibody will go within the fetus, that will also do the lysis of the RBC. As a result, we will see this type of the problem in terms of the erythroblastosis fetalis. And also we can get the autoimmune neutropenia. Now this type of thing we can get in terms of the target cell depletions of the destruction antibody mediated without inflammation. The next part, then class B, we will talk about over here. The class B, this will be complement or FC receptor mediated inflammation. This is the hallmark, and it is written over here, inflammation. That means in type B you will get what? We will get the inflammation here. Okay. Now in this cases, try to understand from here that, for example, we are talking in terms of that we have already said that we will get the autoantibodies against the cell surface or cellular matrix. So for example, in these cases we will get the autoantibodies against the what? Glomerular basement membrane. Basically, we know there is the presence of what? Type 4 collagen, right? Basically, we know within the glomerular basement membrane there is the presence of what? Type 4 collagen. Now, sometimes we see what problem over here, that in these cases there are some portions of the type 4 collagens. Okay, let me show it more clearly for you. Outside these things, for example, these antigens, there are certain types of the antigens. Those are not collagenous. For example, these are the antigens. This is not collagenous. Let me use the red color for it. It's a non-collagenous part. It's the non-collagenous part. Now, if this non-collagenous part or antigens, we can see the productions of the autoantibodies. You can get the productions of autoantibodies. Now, whenever the autoantibodies will come over here, try to understand. At first, they will what do? They will activate the complement system. So, whenever the complement system that will be activated, try to understand from here. So, first what? The one will come, then we will get the C4, we will get then what? C2, then we will get C3b, C3b, then we will get C5b, C6, C7, C8, C9. So, we know these things. And whenever we get the membrane data complex that will form, that will just do the formations of the pore in these cases, and whenever the pore will be formed in these cases, definitely there will be problem. Whatever the problem we will see, try to understand from here. This extracellular and intracellular things that will go outside. One moreover we will get over here. We are getting the productions of the C3. In these cases, we will get the C3 that will break down into C3 and C3b. Now we will get C3a, C5a, and productions of the C4a also. Now C5a that is the chemotactic factor for the neutrophil. So the neutrophil that will come over here. So whenever the neutrophil that will come over here, whatever the neutrophil the problem we will see, whenever the neutrophil will come, they will secrete the different kinds of the destructive enzymes first. Destructive enzymes. Then they can also do what? Oxygen derived radicals. They can also release oxygen derived free radicals. As a result, these situations will be more bad, okay, in these cases. Now, along with that, we will see the neutrophil will come. As a result, the inflammatory cells, the neutrophil that will come over here. Along with that, the C5A, C3 and the C5A, both of them, they will just the chemotactic factor for the mast cell. So we will see also the arrival of the mast cell in these cases, in these regions. For example, this is the mast cell. We will get the mast cell. We will get what the neutrophils will get the complement system. As a result, we will see there is there will what there we can see there is a inflammation. So we can say complement or FC receptor mediated inflammation in these cases. For the classical example, we did talk about in case of the glomerular basement membrane. Basically, we know that type 4 collagen is present over here, but the certain types of the portions where, for example, in this red portions there is a what non-collagenous part, against what we will get the auto productions of the autoantibodies. Whenever the autoantibodies are produced, definitely the FC regions of the antibody that will be activated, and we will see there will be certain types of the complements will come over here. One of them, the C3A, C5A and the C4A, that will be produced also, and these things will just they are the chemotactic factors for attracting the different kinds of the inflammatory cells. So we got over in times of, and this type of the disease that is known as what? Good-Pasteur syndrome. Good-Pasteur syndrome. Now, Good-Pasteur syndrome, we can see in these cases. In Good-Pasteur syndrome, we will get the problem as far as the glomerular basement membrane is concerned. And so this type of the collagen we also get in terms of what? In terms of the alveolar membrane. Alveolar membrane. Okay. In terms of the alveolar membrane, we will get over here that there will be also the alveolar membrane that will be also damaged. There will be also the inflammations. And whenever we are seeing the inflammation, there is a chances for having what? Hemoptosis. Okay, in these cases there is a chances for seeing the hemoptosis. So this kind of a thing we can also see. That means class B, complement mediated. We can get also where in terms of acute rheumatic fever. acute rheumatic fever. In these cases, we can get also what this type of the problem. Basically, in terms of the acute rheumatic fever, for whatever the regions we are getting the sore throat for for the invading of the bacteria, Streptococcus, beta hemolyticus, Lancefield group A. I am repeating once again, Streptococcus, beta hemolyticus, Lancefield group A, that is coming in the sore regions. They are causing the sore throat. After that, that is getting deposited in the different tissues. For example, this type of the antigens or similar types of the antigens will get the will get the presence of the similar types of the antigens like this bacteria where within the endocardium, so over here. So that can cause also the what endocarditis. We can sometimes it can cause also the myocarditis, pericarditis also, and that can also problem with the valve levels. So we'll get the valvular vegetations. It can cause in the CNS, that can cause the chorea. It can cause the some subcutaneous tissues. It can cause a problem in the what in the joints. That can cause the arthritis also. Because the same thing we'll get here, we'll get also the autoantibodies, and the same type of the problem we'll see over here. Now acute rheumatic fever we'll get over here. Now another type, then now type C. What type of the problem we'll get in type C? In type C, antibody mediated cellular dysfunctions. Antibody mediated, let's show the antibody mediated cell dysfunction. Antibody mediated cell dysfunctions. Now how can we just separate them? Let's erase some part from here. Then I think it will be easier for you to have the space in these regions. Okay, now see antibody mediated cell dysfunction. For this thing, the classical example is the Graves' disease. What do we know in these cases? The thyroid gland. And if you see the thyroid gland, within the thyroid gland there are the follicular cells. So if I just show you from here, there are the follicular cells. Now within the follicular cells, we know there is the receptors for TSH. There is the receptors for TSH. Receptors for TSH. That is, TSH will get bind over here, and we will see the productions of T3 and T4. Now, for any region, if we see that the productions of the autoantibodies against the TSH receptors, if we see the autoantibodies against the TSH receptors, so whenever the antibodies, they will get bind over within the TSH receptors over here. What type of the problem we'll see? They will lead to the formations of the they will stimulate the what? Follicular cells. As a result, we will see the productions of the more amount of the T3 and T4, because in the place of TSH receptors, in the place of TSH where TSH is supposed to be bind, by replacing the TSH, the autoantibody is getting bind within the TSH receptors, and they are what? They are activating the follicular cells to produce the more amount of the T3 and T4. And this condition we can consider what? We can consider this condition as hypothyroidism. Hyperthyroidism. We will see the productions of the more amount of the T3 and T4, and you can consider these things in terms of the hyperthyroidism. Hyperthyroidism is on type of the examples. Next type of the examples, where can we see the antibody mediated cell dysfunction in terms of the type 2 hypersensitivity. For these things, let's draw a little bit of this part. Now, from here, basically we know what? Alpha motor neuron, that will come over here. Where? Alpha motor neuron, that will release the what? Acetylcholine, and this acetylcholine, that will get bind with the receptors for the acetylcholine, that is presence of the muscles. Now there is the receptors for what? There is the receptors for acetylcholine. Now normally what do we know? Whenever these things will come over here, they will get bind in these regions, and we will